My name is Sarah Jane Dunn. I'm a postdoc here in the Biological Computation Group at Microsoft Research in Cambridge. And I'm going to talk today about a collaborative project that we're working on with biologists at the University of Cambridge. That's Graziano Martello and Austin Smith at the Centre for Stem Cell Research. So the project is entitled Uncovering the Potential of Stem Cells Through Formal Reasoning, and it's about understanding how stem cells work. So the first question is, well, what are embryonic stem cells? These are a unique type of cell which are derived from the developing embryo, and they arise transiently in the embryo, but they have two very special properties. The first is that they self-renew, and they do so indefinitely given the right culture conditions. But secondly, they're what's called pluripotent, and that's a property which means that they can go on to generate any and all of the adult cell types that you will find in your body. And this makes them a very important type of cell for regenerative medicine, because if we can harness the potential of stem cells, then we can go on to develop treatments for disease and for injury. So this really is the question that the field has to answer. How is it that stem cells make the decision to either self-renew or to commit to a different type of cell? And so what do we already know about stem cells? Well, every cell's identity and its function is defined by the genes that it expresses. And we now know quite a bit about the different genes that are important for pluripotency in stem cells, and how these genes are switched on and off and change to form different adult cell types. But what we don't know is how and why does an embryonic stem cell choose one gene expression pattern that might mean it stays pluripotent over another, which might mean that it goes on to be a heart cell or a pancreas cell or a lung cell. And so here in the Biological Computation Group, we believe that these decisions are made by performing computation, that cells perform information processing, and the subcellular machinery of the cell is actually machinery in the service of this information processing. So the question that we want to answer is, what is the biological program that governs embryonic stem cell decision making? So what we decided to do was adapt formal methods that are traditionally used in computer science to try to uncover this program, to try to look for the essential genes and the essential interactions that form the program that governs stem cell decision making. To do this, we need to move from biological data to logical models that we have the capability to analyze. So we take our data and we have to make a simplifying abstraction. And the abstraction that we've chosen is to assume that genes, well, they're either going to be switched on or off. And then in some way, these genes influence the other genes and whether they're going to be on or off. And so this is the abstraction that we make, which actually means we can encode these models as something that we can analyze. And we're using something called the Z3 theorem prover, which is used widely across Microsoft to verify software, to search that it hasn't got bugs and that it does what it says it does. So this is kind of interesting because we're using this software for a biological system and not for one of our typical computational systems. And so what I'm going to show in this demo is how we've used these methods to adopt a strategy for studying all of the possible programs that govern pluripotency. And so I'm going to flick up this tool now. And so, as I said, there are a lot of genes and a lot of possible interactions that could determine what decision a stem cell makes. And so we have to have some way of searching through all these different combinations to derive what possible programs there might be and to make sure that those programs do what we're expecting them to do. And actually this is a very big problem because it would take you longer than the age of the universe to go through all of these different possible combinations. So the first thing that we have to do is tell the solver which genes we're interested in and what possible interactions there could be between these genes. So first of all, we pass that to the solver, and then we also have to say what kind of behavior are we expecting. And so we create what's called a list of specifications. And those are just the expected gene expression patterns under different inputs or under different gene knockouts. And so we have that kind of information provided to us by our collaborators. And what you can see here is a small summary of some of the solutions that the solver has found so far. Set this off running earlier. Each column is a possible program that is proven formally to capture all of that expected behavior of the stem cell. And so each row in the column is a different interaction, where a green indicates that there's a positive action between those two genes, or red indicates that there's a negative action, so one gene is inhibiting the other. So what you get in this is a summary, first of all, of all the different possible programs that can account for this behavior. But we can actually look at an individual solution itself. So here we can look at the topology of the different interactions that the solver says are needed to satisfy that behavior. We can also look at the dynamics of that particular program to see which genes switch off and when they do so. And this gives us a picture of how these programs are able to satisfy what we're expecting of them. But one thing that's quite neat about this is that we can ask the solver to return us only those programs that have the fewest number of interactions. Because as I said earlier, what we're searching for is the essential program that's governing pluripotency. That means we don't really want any unnecessary interactions. And so using this solver, we're converging towards, well, we're converging away from what's kind of a hairball, a lot of genes, a lot of possible interactions, which we have to unravel somehow, towards a program of interactions that determines whether that stem cell will stay a stem cell or maybe go on to be a different type of cell. Thank you very much.